Hello, sir.
Yeah, hello, madam.
So I made you as a host. You can start the meet.
Okay, just a second. I think I'll try to open my. Thank you, madam. I'm sorry for the disturbance. So I think I was talking about how the drug permeation across the skin can be enhanced for drugs that are otherwise poorly permeable. So there are several formulation approaches. Many people are working on lipid-based drug delivery systems and microemulsions and nanoparticulate drug delivery systems and such things. Of course, the most common approach is the use of chemical permeation enhancers, where you use certain excipients that would enhance the delivery of drugs across the skin by the way of acting in the formulation or by the way of acting on the skin. So I'm going to narrate a little bit about the mechanisms of chemical permeation enhancers. Last but not the least, and there's a lot of research going on in these physical or active methods of drug delivery permeation enhancement techniques. Some are like electrically mediated techniques, and some are ultrasound mediated techniques. Microneedle, of course, is a mechanical way of disrupting the stratum corneum and getting the drugs into the skin. So the chemical permeation enhancers basically are known to act by three main mechanisms. I would say they act either by one or more of the mechanisms together. So the chemical permeation enhancers could be just the solvents that you use in your formulation, or the lipids or surfactants or something like terpenes and such things. Basically what they do is they enhance the dissolution of drug in the stratum corneum. They enhance the partitioning of drug in the stratum corneum, or they act as solvents that would enhance the thermodynamic potential or thermodynamic energy of the drug molecule to get driven across the stratum corneum. Or there are chemical permeation enhancers that would disrupt. They disorganize the organized lipid structures and pave pathways for the drug molecule to permeate across the skin. How does the physical permeation enhancement techniques work? The physical permeation enhancement techniques, for example, like iontophoresis per se, the iontophoresis technique is you use a small amount of current, like a small, very less current density, to drive the drug molecule across the skin, where you are matching the polarity of the electrode with the polarity of the drug molecule, so that there is a repulsive force experienced by the drug ion, so that it experiences a driving force and goes across the barrier. But there are disruptive techniques like sonophoresis per se, and then electroporation. Electroporation is also an electrically mediated drug delivery system. The difference between electroporation and iontophoresis is that in electroporation, you are using high voltage electrical pulses, but they're like short pulses. They're like microsecond pulses. They're not continuously applied. When you apply, it forms pores in the skin lipids, and thus the molecule will be able to go through the pores, and reach the systemic circulation. Sonophoresis is a similar technique where you use ultrasound to damage the stratum corneum lipids. Microneedle is different from all these techniques, where you don't really use any physical energy, except you're applying a physical force to apply the microneedles. So microneedles are basically micron size tiny needles. An array of needles are applied onto the skin, and you disrupt the stratum corneum and deliver the drug molecules. So let's start with the microneedles. with an introduction to microneedles. Microneedle technology is not like a very recent technology. It's about like 2 or 3 decades ago. People have started using this technology for several purposes, mainly in the cosmetic dermatology. So basically, people use the microneedles for treating the wrinkles, and then the burn wounds or the burn scars. When the people have, when the person has a burn scar, you apply the microneedles, and then the appearance of the skin gets better and almost close to normal. Similarly, the wrinkles also was one of the major applications. Basically, this was sold in the name of derma roller. Derma roller is basically a roller with tiny needles on top of it. You just roll it on top of the skin where you want the new collagen to form. So like on the wrinkles and on the burn scars and such things, so that eventually the skin gets better. The mechanism basically is like you apply the roller and you disrupt the stratum corneum and the epidermis, a part of the epidermis, to form channels. Okay, so in these channels, the new collagen is formed, and eventually the skin becomes the skin aesthetically becomes better, appearance becomes close to normal. So in the picture here in the derma roller, if you see this is an enlarged picture basically. Initially, when the derma roller was introduced, they were like selling it for some $800 per derma roller, and eventually the Chinese came into the market and made it like $20. You can get it on Amazon also. So these needles, you can clearly see these needles. These needles were not really micron size. They were like about 1.5 millimeter to 2 millimeters. So then in that case, would that hurt? Yes, of course, it would hurt. Okay, it would bleed. Yes, it would bleed. But this was a cosmetic application. This was a cosmetic, way of treating the scars, as well as the scars, as well as the burn scars, as well as the wrinkles. Okay, then people started thinking like, why not we use this for dermal drug delivery, right? So in case of dermal drug delivery, the idea was, or the approach was, like, if you use a hypodermic needle, right? The hypodermic needle would penetrate deep into the skin. It would pass the stratum corneum, it would pass the dermis, and it would touch it would cross the epidermis, and it would touch the dermis, right? So the thing here is, like, there are no blood vessels and nerve endings in the epidermis, and there are in the stratum corneum or in the epidermis, but there are blood vessels and nerve endings here in the dermis. So when the hypodermic needle goes into the skin, it reaches far enough down to touch the nerve endings and the blood vessels in the dermis. So that's why you feel the pain. That's why you see bleeding after injections and such things. So scientists hypothesized, like, why not we make microneedles that would just penetrate only into the stratum corneum and epidermis? Because once the molecule is released here in the epidermal region, it would eventually diffuse into the dermis and get absorbed into the systemic circulation, right? So if the microneedles are, if the needles are just enough to penetrate into the epidermis and not reaching far enough down to dermis to touch the nerves, then it could be a pain-free way of delivering drugs, right? The main barrier is stratum corneum for polar molecules. The main barrier for lipophilic molecules is this epidermis, right? So if you can make a microneedle system, you would be able to deliver both hydrophilic as well as lipophilic molecules deep down into the skin, either for dermal application or for transdermal delivery. So this was the thought initially, and this was, like, mainly envisioned as a pain-free way of delivering drugs, mainly for vaccine delivery, as many people are needle phobic, as you see. Particularly kids are needle phobic. They don't like injections, and it's really painful, and people cannot take too many injections within a short span of time. Like, if you have to take, like, for example, if you're suffering with iron deficiency anemia, you would have to take iron dextran injection. That is like the most painful intramuscular injection, and you would have to administer it frequently, and it's not really comfortable. So in such unique applications, the microneedle would come in handy. So this was the initial thought, and people started looking at microneedle as a potential transdermal drug delivery system for delivering hydrophilic molecules, lipophilic molecules, and even macromolecules, like as I said, vaccines and peptides and hormones and such things. So that's how the research started. And then when people started making microneedles, like, how do you know if these microneedles that you make, they're like very tiny, right? They're like what, 200 to 500 micrometer in size. So you make such tiny microneedles. You make an array of it, like, the microneedles are in a small patch. It's about 100 microneedles within about half square centimeter, right? So you apply it onto the skin. How sure are you that they're disrupting the skin? How sure are you that they're delivering the drug into the, inside the inner layers of the skin? So to test that, we need to have some kind of a tool, right? So generally, to test the integrity of skin, when we set up skin permeation testing, we use two different types of tools. One is the transepidermal water evaporation rate, and the other one is transepidermal electrical resistivity. So these are the two tools that people use to find out if the skin is intact or not. This is very useful when you're setting up an in vitro permeation testing, because you don't want a ruptured skin, right? So for your permeation studies, so you would want to set up one of these to find out if the skin is intact and good for your permeation testing. My standard, from my experience in my lab, what we see is, like, if the skin is of resistivity of more than 10 kilo ohm centimeter square, or a transepidermal water loss of less than 10 grams per meter square per centimeter square, then per meter square, sorry, then the skin is good for permeation studies. So here we are using these tools to find out if the microneedle really ruptured an intact skin and made pathways for the drug molecule to diffuse across, right? So what is this TWL? TWL is quite commonly used because of its convenience, and because of the handheld machines available, like this vapometer from Delphin Technologies and such things. Electrical resistivity is a bit complicated, because I worked in a biophysics research laboratory at the Russell Park Cancer Institute during my postdoc studies, and I kind of have an idea about how this electrical resistivity equipment works, and I've set up my own at the research lab. I had in the University of Mississippi, and now at my contract research center, as well as at IDBR. So let's talk more about what is this TWL is. TWL is basically you're measuring the rate of evaporation of water across the skin. Why is there always an evaporation of water from inside to outside? Because there is a lot of water in the inner layers of the skin, and there is hardly any water in the stratum corneum. So this creates a concentration gradient, right? Because of the concentration gradient, the water always evaporates. It always, propagates from the inner layers to the upper layer, and it keeps, there's always an evaporation taking place, and intact skin has a certain value of TWL value. TWL magnitude. Okay, so when you rupture the skin, when you rupture the stratum corneum, this TWL value will increase. Okay, so this is the main measure for finding out if the stratum corneum is being ruptured by microneedles or not. And this is also one of the tools to measure, like, how long will it take for the skin to recover? Because once you make a pore, okay, people imagine that, or speculated that the pores would remain open for quite a long time, because you're kind of rupturing the stratum corneum mechanically. But it's not so. We have tested this, and we have found that, this is the TWL value of intact skin. Okay, and then when you rupture it, when using the microneedle that we prepared, okay, the microneedle that we designed led to an increase in the TWL value, immediately after we rupture the skin with microneedles. And then eventually the TWL value will come back to normal. Okay, so within about 6 to 12 hours, you can say all these spores are reformed. Okay, so it depends on what kind of microneedle, what shape microneedles, what pitch distance is designed, between the microneedles and such things, on the level of rupture and the time taken for recovery of microneedle, recovery of micropores passed in the skin. For example, if you have too many microneedles within a short, within a small surface area, okay, then you experience what is called as a nail bed effect. So this, in this nail bed, the child is sleeping on a nail bed but experiencing no pain. So this is all like full of nails, but the nails are placed so close to each other that they're not able to hurt the child. They're not able to apply enough force onto the skin to make the child perceive the pain. So the same thing happens if you have the microneedles too close to each other. Then it will not be able to penetrate or apply enough force to rupture the stratum corneum. So the point that I'm trying to make is whether the microneedles will penetrate the skin, whether the microneedle that penetrates the skin forms pores, that are like deep driven or larger in dimension, whether the microneedles would, the micropores form would recover. If they recover, how long will it take? All these things depends on the design of the microneedles and the array. So, so basically now we are at a stage of um, introspecting, like why are we really giving so much emphasis on this technology, microneedle technology, when there are so many physical techniques and chemical techniques of enhancing the permeation of drugs, right? So compared to any other physical technique, the most efficient technique that can rupture the stratum corneum is microneedle. So it can perturb the stratum corneum, and it is useful for delivering both polar molecules and non-polar molecules, and even the large molecules, right? And you can also deliver the drug rapidly into the stratum corneum, into the stratum corneum as well as the epidermis. And sometimes you may have to deliver certain excipients, functional excipients that would enhance the activity of the API. Okay, for example, in case of vaccines, you would need to deliver the adjuvants, right? So we had a project in which we tried to deliver CPGs along with certain vaccines as adjuvants. So they're all huge molecules. So none of the techniques that are being investigated under the domain of physical permeation enhancement can do an efficient job, as efficient as microneedles. So there are different types of microneedles. There are something called as halo microneedles. The halo microneedles are even being used for injecting the liquids into the skin. So this is also an amazing technology that I'm going to talk about in a little bit, in little bit detail. So basically, there are different types of microneedles. The types of microneedles include hollow microneedles. There are solid microneedles. In the solid microneedle domain, you have microneedles made up of metallic materials, which are either used for pre-treatment or used for delivery of drugs. Basically, you're coating the microneedle. The surface of the microneedle is coated with the API that you would want to deliver, and you use the metallic microneedles to deliver the drug, and you will retract the microneedles later after the delivery of drugs. And there is another very promising and upcoming area, polymeric microneedles, where you load the API into the polymeric matrix and deliver the drug. And sometimes the API is loaded into a carrier, like your nanoparticles or liposomes or neosomes or anything of that sort. You can incorporate the entire carrier in the microneedle and deliver the microneedle, deliver the entire carrier with the API into the skin. So let's just look at few examples of all these different types of microneedles. Hollow microneedles. Of course, I've not personally worked on this hollow microneedles. I have come across on 3M's hollow microstructure transdermal technology. It is called as HMTS technology. So they have a device into which you load the cartridge that you want to. The cartridge contains the solution that you would want to deliver into the skin. So you see the picture of how the microneedles look like, the hollow microneedles look like. So you place the entire device on top of the skin. These needles would penetrate into the skin, and you apply activate the actuator to deliver the liquid, and they claim that up to 2 milliliters of the drug solution can be delivered into the skin. And I think these microneedles are pretty long. Also, they're like about 1.5 millimeter microneedles, and then you see the transverse section of the skin after application of the microneedle, the hollow microneedle. So you can deliver in a quite large volume into the dermis using this device. And this is the anatomy of the device. Is basically a hydraulics based device. And 3M also has a pilot facility to manufacture, if you would want to manufacture the microneedles, these kind of devices for clinical trials and such things. So they do have a GMP facility as well. So moving forward, I will start with the solid microneedles. The solid microneedles are basically um, microneedles made up of either metals or polymers. I'm starting with the metallic microneedles. What you see here is called derma stamp. This is about 0.5 millimeter. That's about 500 micrometer microneedles. So we use these microneedles basically as a pre-treatment device. So meaning that, before you apply the formulation, you pre-treat the skin with these microneedles, open up the skin, make micropores, and then apply the formulation on top of it and see, if it would enhance the delivery, right? So we tried different solvent systems. So our objective was to see like how the physical chemical properties of the solvent would influence the delivery of drugs across the pre-treated microneedle skin. But we didn't see a big difference between the solvents. Basically, it was a convective transport. What you see in the blue is the passive permeation of drug. So this profile is a bit different. If you see the way we have represented the profile, it's like flux versus time. The flux is nothing but the rate of drug permeation between the two time points. You see that initially the flux is higher even in passive, and then eventually it would drop down. And then in case of microneedle pre-treated, you see a lot higher permeation flux. And then you see that within 3 hours or so, 2.5 hours, you see that the rate of permeation would almost come down to normal. So meaning that within 3 hours, the pores that are formed are not open anymore for the solvent to diffuse into, or the solvent to deliver drug by the process of convective transport. So we tried different solvents like ethanol, which is supposed to be a very volatile solvent, and then we tried out water. So this like all these solvents were like about up to 3 hours were able to deliver the drug. In this case, triacetin, same thing, and then a triethyl citrate was something that we found that it could deliver, it could prolong the pore lifetime of the pore for quite long time. Okay, so when you use triethyl citrate, the pores remained open for a long time, and we were able to get a lot of drug across the skin. So this was the whole objective. So we initially hypothesized that the solvent properties like density and polarity, dipole moment, and such things would be the main factors that would determine the extent of delivery of drug by convective transport. But it didn't turn out to be what we expected. By it ended up showing that when you use triethyl citrate and so many other solvents that I'm not able to disclose here, so many other solvents, when we use it increased the lifespan of the micropores, not like days together, but by a few hours. If you imagine, like if there are pores existing, and if you can prolong the lifespan of the micropores even by 30 minutes or 1 hour, you should be able to get a lot more drug right into the skin. So that was the whole objective of this entire experiment, right? And then, as I mentioned, these metallic microneedles are also used for delivering drugs directly, meaning that you coat the drug, you coat the antigen, you coat the vaccine, okay, or the peptide or hormone or whatever you want to deliver on top of the solid microneedles, okay, and then you inject it you apply it onto the skin, and all the contents that are present on the surface will be released into into the extracellular fluid of the skin, and then eventually you would retract the microneedle, the metallic microneedle, after delivery of drug. So one such kind of patch, microflux, was developed. I think Zosano was the company that initially started with this. These are titanium made, patches. Basically, this is the patch with the glue around, with an adhesive around, and these are the projections. You can see the projections here. These are the titanic microneedles, and then titanium microneedles, and then so these titanium microneedles are coated with the antigen, and then there is an applicator. You apply it onto the skin, and then you remove the applicator. The patch remains on the skin until all the contents in the microneedle, all the contents that are coated on the microneedle is delivered into the skin. So this is one such solid metallic microneedle application that's been very successful, and people have been using it. People have been researching it more and more, and I think it's in clinical trials right now. So there are so many other applications of this metallic microneedles people have explored, or like we did in this case of angular delivery. So what is angular delivery? The delivery of drugs into the nail plate. You know how hard the nail plate is. Nail plate is full of dead cells filled with keratin. There is no lipid, hardly. There's about 1% lipids. The molecule cannot diffuse across the nail plate easily. So why do we want to deliver the drugs into the nail plate? Because the nail plate is prone to fungal infection. Okay, and then condition called psoriasis also. So there are two major nail disorders that are seen, particularly in the Western world. You see onychomycosis, the fungal infection of the nail. What the pictures, what I showed here are the fungal infection of the nail. So what is this fungal infection of the nail? The fungus T. rubrum, Trichophyton rubrum, affects the nail plate and the nail bed and the nail matrix. Okay, this is the nail matrix. This is the transverse section of the nail of the toe. Okay, this whole entire thing is called as nail apparatus. In the nail apparatus, you see this nail plate and the nail bed. Nail bed is a soft tissue. Nail matrix is the place where the nail originates, right? So the nail is formed here and it is pushed forward. Okay, and then the fungus goes into the matrix and into the nail bed. It's very hard to deliver drugs into this region. The current treatment is oral delivery of antifungal drugs, and it's huge doses of antifungal drugs for about 8 months to 1 year. That's a lot of antifungal drug to give it to a patient, because many of the antifungal drugs are associated with severe gastric and hepatic and systemic side effects. So we want to deliver drugs into the nail plate and into the nail bed locally, rather than systemically. So we explored many different ways of delivering drugs by using iontophoresis, by passive techniques and such things. So one of my students was interested in looking at delivering drugs by puncturing the nail plate with the microneedles. So this puncturing nail plate is not a new technique. People have been using very sophisticated techniques like laser drilling technique and such things. Very sophisticated techniques, and like very expensive techniques. Like you have to apply a very small, very tiny jet of laser to drill holes in the nail plate and then deliver the drug into the nail plate. But we thought, like, why not we go for a very simple way of rupturing the nail plate, and we created pores using the microneedle. So these microneedles were just enough, the length of the microneedles was just enough to drill a hole only in the nail plate, not reach the nail bed, because if it reaches the nail bed, the patient would perceive the pain. Okay, so you have to design the microneedles in such a way that they penetrate only through the nail plate. And then when we see this, the picture here, the transverse section shows the pore that we created. This has gone only through the nail plate. And then once you apply a angular formulation, maybe a solution or a gel, okay, you will be able to deliver a significant amount of drug into the deeper layers, the subungual layers called such as nail bed and the nail matrix. So we were not only able to deliver a significant amount of drug into the nail bed, but also we were able to deliver the drug into the nail plate in the peripheral region. Okay, so other than where we applied, where we have created the pores, the drug is able to go, diffuse laterally as well. So this was one interesting application that we tried with microneedles. So this is about the solid microneedles, right? So let's look at the polymeric microneedles. So we completed hollow microneedles, the solid microneedles, and that the metallic, where we use pre-treatment, where we use metallic microneedles for pre-treatment, and then we looked at a case where the drug was coated on the metallic microneedles to deliver into the skin. So let's start with the polymeric microneedles for dermal application. They're also called as dissolving microneedles or disintegrating microneedles. Okay, so polymeric microneedles for dermal and transdermal delivery systems. So these basically, what you see here is an electron microscopic picture of a polymeric microneedle. So this polymeric microneedle is about 500 micrometer in length. You can see the structure. They are like pyramidal in nature, and the base is about the base of the microneedles. The base is about 250 micrometer. The tip is about 25 micrometer. So only if it is like 25 micrometer or less, it will be able to penetrate into the stratum corneum. If it gets blunt, then it is hard to make it go into the stratum corneum. So of course, the entire needle should be of sufficient mechanical strength to be able to rupture the stratum corneum. So these microneedles are made up of polymers that would dissolve or disintegrate rapidly, so that you will be able to deliver the drug instantaneously into the skin. For example, you see here, so this is the picture of the first picture is of the array of microneedle. So the array of microneedle, this is about like 0.5 square centimeter with about 100 microneedles present in them, and the drug loaded. And then when you insert them into the skin, after a few minutes, you see they deform and they change the shape. And after within like 3 or 5 minutes, you see this the entire microneedle, the polymer would dissolve and it would become like this, like planar. So at this time, you can remove this base plate, and the polymer is inside the skin. It will disintegrate or dissolve and release all the drug. So this particular technology has been utilized for delivery of several drugs. People have been working a lot on antiviral drugs and several other medications. Initially, we had collaborated with Dr. Ryan Donnelly at the University of Queen's University, Belfast, to make microneedles for delivery of iron compounds, mainly to overcome the pain associated with the iron dextran injection. We have published a couple of papers in Journal of Pharmaceutical Science also. So we did a lot of work in the space of anemia. And then we tried different applications also. One of my students, Dr. Apurva Panda, worked on delivery of nicotine. So basically, when you want to, the treatment is to stop smoking, right? So nicotine is available in the form of patches, chewing gums, and different kinds of formulations, mainly for the people to stop smoking, to make up their mind and stop smoking. So what is required here? So whenever the patient, whenever we cannot call him a patient, whenever the subject feels like smoking, he should have enough nicotine in his blood so that he can circumvent that thought, right? So the problem with the current delivery systems like chewing gums and patches and such things is that there is a long lag between the time you apply it and the time you see a surge in the drug concentration in the blood. To overcome that, we made microneedles, and we found that when you apply microneedles incorporated with nicotine, we were able to deliver significant amount of nicotine into the systemic circulation. So what you see, the black line is the IV plasma pharmacokinetics of drug nicotine, and then the red line or the brown line is the plasma concentration time profile of nicotine administered by microneedles. So it looks like the microneedle is an excellent technology for making nicotine patches to overcome smoking or smoking cessation rather. So this is one of the research done by my research student, a PhD research student, Apurva, and we published in Journal of Drug Delivery Science and Technology. And then we also use this solid microneedle or polymeric microneedle for controlled release delivery system. So you all know that anytime we talk about controlled release drug delivery system, particularly nanoparticles and microparticles and biodegradable polymer, the first thing that comes to our mind is PLGA, right? So PLGA is one of the approved polymers. So we prepared PLGA microneedles. Just the plain PLGA microneedles won't work well, didn't work well for us. So we had to combine it with PVA. So different amounts of PVA was incorporated into PLGA, and we prepared the microneedles to see if we can apply the patch onto the skin and have a controlled release of drug, right? So we also looked at the effect of PVA on the release profile. We used just a FITC dextran 4 kilodalton. This is like considered a macromolecule, right? The 4,000 Dalton molecular weight is considered a macromolecule. So our objective was to design a drug delivery system for controlled release of a macromolecule. So we use 4 kilodalton dextran as a model macromolecule, I would say. And then we found that when you increase the PVA concentration, the rate of release would increase. You can see the time taken for the drug to attain a significant percentage of release is up to 72 hours. So we use different grades of PLGA, and we could accomplish a prolonged delivery. And unlike in case of hyaluronic acid or polyvinyl pyrrolidine based microneedles, in which you deliver the drug instantaneously, immediately within 5 minutes the polymer is dissolved and the drug is in the skin. Unlike that, in case of PLGA microneedles, we were able to deliver the drug over a long period of time. So so far what we learned in dissolving or disintegrating microneedles, the polymers and the other excipients that you use for making the microneedles should be biodegradable, it should be FDA approved, it should be biodegradable, it should be biocompatible and such things. And there is a possibility of delivering drug instantaneously or in a controlled manner. So sometimes if you deliver instantaneously, that could lead to potential skin irritation and such things, depending on the nature of the API and the excipients, right? And then there are cases where you may have to deliver the polymer and the other excipients present in the microneedle unnecessarily into the tissue, right? So they may not be really required. Like every time you apply a microneedle, you are delivering a significant content of the polymer, maybe a biodegradable polymer, but like for example, PVP is not a really biodegradable polymer. Still, you would end up delivering quite a significant amount of PVP into the skin. Imagine if you have to make a larger size microneedle patch with PVP. So that leads to a significant amount of PVP being loaded into the body. So so we were thinking, like, why not we design a microneedle system in which we only deliver the drug and not the polymer. Okay, not the content, not the excipients, just the drug gets diffused across, like a transdermal patch, right? You have so many things in transdermal patch, but when you apply it, it is just the API that goes across the skin. Maybe sometimes some permeation enhancers like propylene glycol and such things might be able to penetrate, but not really the polymer and the adhesive and such things. So we wanted to deliver something that allows the diffusion of API alone and not the polymer and the excipients. Mainly for delivery of drugs, like per se, if you want to deliver drugs into the back of the eye. Okay, there are so many non-dermal applications of microneedles these days, right? People are using microneedle for delivery of drugs into the eye, particularly into the back of the eye, because it's challenging to deliver drugs into the back of the eye. Suppose if you want to treat uveitis and retinal infections and so many other back of the eye problems, or into the sublingual tissue per se, as one of our researchers is working right now on the sublingual delivery of drugs. So you don't want to deliver a lot of excipient because it will eventually reach very sensitive tissues, right? So we don't want any complications because of the delivery of polymer. So in that direction, we thought about making polymer coated polymeric microneedles. So PCPM is what we short termed as. So basically, what this is, you have a microneedle, a core microneedle, and there is a coating around, right? So this coating is basically a semi-permeable membrane, right? There is a base plate, and there is a microneedle, a core microneedle, like the microneedles I showed you in the pictures in the past. And then this is the coating, okay? So you can incorporate the drug in the coat in the core, so that the drug would once you put it into the tissue, there is an equilibrium established between the core microneedle and the extracellular fluid outside in the tissue, and the drug would diffuse along the concentration gradient into the tissue. You can formulate this microneedle in different ways. I called this as type 1, type 2, type 3. Type 1 is drug in core only, and then you can call it, you can make like drug in core and coat. If you want to deliver two drugs that are not really compatible with each other but need to be delivered simultaneously, you can have a drug in the coat and another drug in the core. And of course, you can have the drug in the base plate as well. So any of these things are possible. So this is what we envisioned to start with. And then we prepared microneedles. We prepared microneedles by the regular mold casting technique, and then the coating was done by dip and spin technique, where basically the microneedles were dipped into a suitable polymeric solution, and then we made the microneedles with a dye to make sure that the polymer coating is uniform across the needle. And then we also tested them for its ability to rupture the skin and such things. I'm going to show you some more data in the future. So basically, when we made these microneedles, we used ethyl cellulose as the coating polymer. So how will you make sure that it is coated intact, right? So how will you make sure that there are no pores in the coating? The coating is uniform and intact. So we incorporated a dye, Nile red, into the coating polymer. This is a plain microneedle. We coated with 1% ethyl cellulose, and then 2%, 3%, and 4%. We found that the 4% was excellent. It was coating the entire microneedle, and it was forming a thin layer around the microneedle. So you can see the difference between the uncoated and the coated microneedles in the electron microscopic picture. This picture, the first picture is the uncoated one. The second picture is the coated one. You can see you can appreciate the difference in the texture. The texture is pretty smooth after coating. And just for your understanding, I just broke a microneedle and took a picture to show like how the coating looks like on top of the microneedles. So once we were sure that we can use 4% ethyl cellulose to coat around, I think this polymer, the core microneedles was prepared with PBP, and then it was coated with ethyl cellulose. Main reason we selected ethyl cellulose was like it should be water insoluble, right? So ethyl cellulose is water insoluble, but it's a semi-permeable. It's hydrophilic, but it does not dissolve. Okay, so it's a semi-permeable membrane. So we use this for delivering drugs, and again we used a dextran, 30 kilodalton dextran as a model molecule to see if we can deliver the drug, if we can play with the design variables to get the desired release. So you see, this the first graph shows like how the uncoated microneedles and the coated microneedles differed. In case of uncoated microneedles, you see that the drug was instantaneously released, but in the coated microneedles, it was kind of retarded. And then we can change the concentration of the polymer that you use for making the core microneedles. You can change the grade of polymer that you use to make the core microneedles. And then you can even play with the grade of the coating microneedle, the coating polymer, right? So like this, you have different things that you can play with to achieve the desired rate of drug release from your microneedle. So the other polymer that we tried was aggregate E100, or this is also for the reason that it is not soluble alkaline pH range. It is soluble only in the acidic pH range. This is an excellent polymer. So what you see in the picture, in this picture is the polymer microneedle coated with aggregate 100 E100, and of course we incorporated a dye just for good microscopic visibility, and we could achieve a good control over the drug release up to 3 hours using this aggregate microneedles. And we used EPO and 100 E100. So both are chemically similar, but it's just available as powder and solution. We use different concentrations to see, this was like a preliminary experiment for us. So just to give you an idea about like how would it look like when you coat the microneedle. So this was the original picture. I kind of did a bit of a photoshopping on this to show you how the core microneedle is embedded within this coat. You can clearly see the coat, right? The border, right? So this is intact after 6 hours of release studies. So after dipping these microneedles in the buffer for about 6 hours. So this is not the fresh one. So even after 6 hours, we see this coat being intact and being acting as a semi-permeable membrane and letting the drug release at a slow fashion. So after all this, we need to make sure that this microneedle would perturb the skin also, right? After coating, you are changing the morphometrics of the microneedle, right? So whether these microneedles are able to perturb, again, we go back to our tool, TWL, transepidermal water loss. All these are coated microneedles, so ethyl cellulose coated and aggregate coated microneedles. When we apply these microneedles on top of the skin, we were able to achieve a significant perturbation of the barrier, skin barrier, really leading to an increased TWL value. So this is a good sign. We are able to coat the microneedles. We are able to preserve the micrometrics that would allow the microneedles to penetrate into the skin. We are able to achieve a prolonged drug release, and we are able to achieve our main goal of not letting any of the polymers and excipients go into the tissue and just allow only the API to diffuse into the tissue. So the other application of this microneedle, the coated microneedle that we were thinking of, was to sample the analytes and substrates from the skin tissue. Like, the two-compartment model, basically, right? So when you deliver the drug into the systemic circulation, the drug would equilibrate itself between the plasma, which is also an extracellular fluid, basically, between the plasma and the extracellular fluid of the skin tissue, right? So there is an equilibrium between the ECF levels of free drug and the plasma-free drug. If you are able to sample the concentration of drug in the ECF, if you're able to measure the concentration of drug in the skin extracellular fluid, you probably would be able to back calculate the amount of drug present in the plasma. So this is one application. The second application is, suppose if you're treating some kind of a skin disorder, right? The amount of free drug present in the skin would become very important, right? You may be loading a lot of drug systemically, but whether the drug is getting into the extracellular fluid of the skin in effective amounts is what really determines the efficacy of the treatment. So you would want to learn about the dermal kinetics of drug, the rate at which the drug would enter into the dermis, the duration up to which the effective concentration of drug remains in the skin, is very important. It's crucial in determining the frequency of administration and the efficacy of treatment, the dosage, and several things. So we don't have great tools to find out how much of drug is there in the extracellular fluid. There are some methods like blister fluid collection and biopsy sampling and such things. They are all invasive methods, and they don't give you any idea about the amount of drug that is present in the unbound form, because only the unbound drug is active, right? The bound drug is not going to be effective. So we thought, why not we use this coated microneedles in the skin to sample the drug into the microneedles. So basically you're making plain microneedles. So the coat microneedle and the core microneedle and the coat microneedle, they don't have any API in it. The drug is already there in the skin. You apply not only skin, any tissue for that matter, right? You apply these microneedles into the skin or into any tissue, the drug that is present in the extracellular fluid would diffuse across the barrier and get into the core, okay, the core microneedle, which is more hydrophilic, and the drug and the polymer that you use should have an affinity to the drug molecule as well. So I'm going to talk a little bit more about the advancement in this technology that we tried. So basically, when you apply these microneedles for sufficiently long time, you would be able to sample the drug from the dermal extracellular fluid. That would help you in determining the exact amount of drug that is present in the extracellular fluid. That in turn would help you in knowing the entire dermal kinetics of drug after whatever route of administration you consider, oral administration or parenteral administration or anything. So we tried again some model molecules. All these are like proof of concept data. We used sodium fluorescein, we used prednisolone and nicotine, and then we tried to sample. So all these drugs were previously loaded into the tissue or into the substrate, and then we applied these microneedles, and then we removed the microneedle after a certain period of time and looked at how much of drug has come with the microneedle. So we found that there was a lot of sodium fluorescein compared to the lipophilic drugs like prednisolone and nicotine. This is quite obvious because the polymer that we used, in the core was PVP, and PVP is quite hydrophilic in nature, and it looks like there is diffusion of hydrophilic molecule. There is selective diffusion of hydrophilic molecule into the core microneedles when you use PVP as the core microneedle. So then we improvised this microneedle technology by incorporating adsorbents. So these adsorbents are great in absorbing lipophilic molecules. So what you see here in the background is an array of microneedles with adsorbent incorporated. Adsorbents are present inside. If you can see small spherical adsorbents present in them. So one of the microneedle is enlarged here. You can see the adsorbent, and then the microneedle, and then the coating on top of it. Something like that is represented schematically here. So again, the same mechanism. And then when we tried the same experiment, we found that the sodium fluorescein was like almost the same. The amount of sodium fluorescein that we could sample was almost the same. It was pretty reproducible also if you look at the error bars, right? But we were able to sample a lot of prednisolone and nicotine and lipophilic representative lipophilic molecules by the use of these adsorbents. So this is an ongoing work. This work was done by Diksha and Diksha at University of Mississippi, who was my master's student, and now she's a PhD student. And this was a part of the work. A significant amount of work was done by Anusha at IDBR as well. And then not last but not the least, one non-dermal application of microneedle that I would like to talk about is the sublingual delivery of cardiovascular drugs. Again, this work is entirely of Anusha, who's a research scientist at Institute for Drug Delivery and Biomedical Research. This girl really mastered the technology of microneedles, and then she developed her own project. This was her own idea to deliver drugs sublingually, basically the cardioprotectant drugs, particularly something like nitroglycerin and isosorbate dinitrate, and some of the cardioprotectants, cardiac stabilizers and such things. Mainly the drugs that are used in treating the cardiac arrest, because there are so many sublingual dosage forms like sublingual tablets and sprays and such things available, but still about 40% of the patients are known to die because of the lag in absorption of drug from all these different formulations, and rapid, as rapid as possible delivery of drug into the sublingual tissue and into the heart is likely to save many more lives. So that was the hypothesis, and that was the objective with which she started. She made this was funded by BIRAC under the Biotechnology Initiative Grant or Ignition Grant. So she incorporated nitroglycerin into this, and and she prepared the microneedles and tested in vitro and in vivo, and she got some very excellent results. And this is one of the previous data that we had generated using sumatriptan. Sumatriptan delivered by intravenous injection, sumatriptan delivered by our microneedle technology. They were leading to almost the same level of plasma levels of drug in rat model. Basically, in the sublingual solution did not deliver as much as all these other approaches delivered. So this is one of the ongoing projects right now, sublingual delivery of microneedles. So um, I would like to conclude by saying that microneedle drug delivery technology is definitely a promising technology. There are several dermal applications, but there are several upcoming emerging non-dermal applications as well for the delivery of drugs into cutaneous tissues, into the nail, into the mucous membrane, sublingual, and several other applications are coming up. So there is going to be a plethora of opportunities in this area. And some of the new approaches that we worked on, like the polymer-coated polymeric microneedles and such things, are really of help when you are trying to deliver drugs into sensitive tissues. So Diksha is also working on using these polymer-coated microneedles for delivering voriconazole into the back of the eye. That's a great application for treating fungal infection. Otherwise, getting enough voriconazole into the back of the eye is really challenging. So, I conclude by saying that this is definitely a great area to explore for younger researchers and aspiring scientists. This has plenty of applications, and we are also learning a lot as we go. With this, I think I would conclude by saying thanks to some of the very important people, my mentor, Dr. Howard Maybach, Dr. Srinath, and Dr. Vijay, and Dr. Srinivas. All these are associated with my company, TPT, in Oxford, Mississippi. And I would also like to thank my graduate students. All these people worked on microneedle technology in one or the other domains. Dr. Naresh, Abhishek, Abhijit, and Diksha. And then the entire team of IDBR at Bangalore, Dr. Shivkumar and Dr. Sarsija, you're going to listen to her in one of the sessions in this symposium. Dr. Vijay, Dr. Pranjyoti, and everybody, Anusha, Pratima, and everybody who's working with us at IDBR. So I would like to thank you all for your patient listening. I know there were some technical glitches. I apologize for them. I would be happy to answer any questions if there is enough time for the questions.
Thank you, sir. So it was a very excellent session, and we are getting the same remarks from all the delegates. That session was very excellent. There's one question from Simran. So she's asking about, briefly explain the formulation procedure of sublingual microneedles.
Formulation of sublingual microneedles.
Microneedles, yes.
Okay, yeah. What would I say? The method of preparation of microneedles, sublingual, or any other any other application is similar. Basically, we use the molds, and then we prepare the polymeric solution. Generally, hyaluronic acid or PVP is the one that we commonly use, but we used cellulose polymers as well. And then you load the mold and centrifuge, and then allow it to cure. There are different ways of doing it, and this is the way we are doing it. And then allow it to cure, and your microneedles are ready. And then eventually, when you want to coat it, you have to design some kind of a spinning instrument. You have to dip the microneedles into a coating solution and spin it. You have to optimize the spinning speed. You have to optimize the coating solution concentration. You have to optimize several things to be sure. It's very tricky to get a uniform coating around the microneedles, because when you coat a tablet, it's a different thing, right? You have a bigger object to coat, and you will know if it's intactly coated or not. But this is like tiny objects, like tiny needles. So unless you look under the microscope, unless you do something like a scanning electron microscopy and such things, you wouldn't know if the coating is properly done or not, uniformly done or not. So this is basically in a simple way to talk about the way we formulate. But I think when you do it, it is not as simple as I narrated it.
Thank you, sir. There's one more question from Sayani Bhattacharya. His question is how the application will be carried out in case of sublingual delivery of microneedles.
Well, sublingual delivery, sublingual microneedles are not any bigger than a sublingual tablet or a sublingual film. Okay, so the subject just need to take the microneedle and stick it under the tongue, like any other tablet. It's basically like a tablet. So the patient wouldn't perceive any pain. So the patient wouldn't know that the microneedle penetrated. Microneedle would just penetrate and do its job.
I hope sir has answered your question. The next question is how to estimate the amount of drug loading into the microneedles?
Well, that is again a thing that need to be standardized. You will incorporate the drug into the polymeric solution. If you are making a polymer-based microneedle, and then after you make the microneedles, you can calculate based on the volume of the microneedle. The dimensions of the mold, the density of your solution, the amount of drug that you incorporated into the solution. You would be able to calculate basically, theoretically, how much of drug should be present in the microneedles. And then, of course, in every batch, like how you test the tablets, per se, you're making 1 lakh tablets, you would take out 20 tablets and test for the drug content. You would do the same thing with the microneedles as well. You have to scrape out the needles and look at the amount of drug present in that.
Thank you, sir. So if there even anybody who wants to unmute yourself and ask question to sir also can be done. Otherwise, we'll end the session. So to summarize today's session, the session was very excellent. It was very nice and a very pleasant session what has been taken place. So started with different approaches of skin permeation and enhancers, and we also came to know the different types of enhancers and different techniques for enhancement, physical techniques. The most interesting thing what I felt was TEWL approach or mechanism to study the permeation of the drugs. And then we studied about the dissolving microneedles. We also came to know about the polymer coated polymeric microneedles. And the most interesting part was about the sublingual delivery of the drugs through the microneedle technology. So it was a very excellent session. I hope all the delegates who attended this session enjoyed it. Over to SP Remat sir to conclude the session.
Thank you, sir. The session was very informative, and especially the polymeric dissolving microneedles as well as polymer coated polymeric microneedles was very interesting. And I thank once again Murthy sir for his excellent lecture. Thank you, sir. I thank all the delegates for their patience listening. Thank you. Thank you.
Thank you for the opportunity.
Thank you, sir. Thank you, sir, for sharing your expertise. So we shall end the meeting now, and we shall rejoin at 2:45 p.m. The next session will be at 3 p.m. by Giriraj Kulkarni, sir. Thank you. Thank you, one and all.
Ma'am, can you repeat the timing, please?
2:45. We'll start the session at 3 p.m. Kindly join at 2:45 so that we'll start the session at 3 p.m. exactly.
Okay.
Okay. Thank you so much.